Let me go ahead and pull up the intro course. Good morning, good afternoon, and good evening. Welcome everybody to our second installation of the ISCBacademy Industry Series. Today I'm very excited to be able to introduce Dr. Max Salick, who is coming from insitro to share with us a talk about single cell resolution image-based CRISPR screening at druggable genome scale. And Max is originally from the engineering field. He has a BS in engineering mechanics and astronomics and a PhD from the material sciences program, both from the University of Wisconsin-Madison. He took his engineering background with him to the complex discipline of neurodevelopmental disorders, and then completed his postdoctoral training in the neuroscience department at the Novartis Institutes for Biomedical Research, building a single-cell RNA-seq platform, conducting a wide array of CRISPR screens, and utilizing stem cells to understand the molecular underpinnings of these diseases. Following his training, Max joined insitro as one of its founding stage scientists, and now leads a multidisciplinary lab focusing on building and establishing new technologies that feed directly into insitro's drug discovery engine. We're so excited to have you here today, Max. For the attendees and for those viewing the recording, if you'd like to catch the next ISCBacademy talk, that will be on September 22nd, where ISCBACademy will be presenting from AstraZeneca, and registration will be up live on the ISCB site shortly. And without further ado, Max, the floor is yours.
All right. Thank you so much, Kristen. Thank you for the introduction. And also thank you for inviting me to come and speak. And thank you, Seth. Behind the scenes, making sure everything's working swimmingly. I want to give a shout out also to our friends over at Bio-Rad that made this connection to begin with, Trey and Eli. And I also want to thank all the people, of course, that have contributed to this project. I'm presenting, but this was the work of, a ton of folks and an incredible team of scientists. So I'm pretty excited to kind of present all their hard work and kind of, showing this platform to the ISCB. All right, so I'm going to start with kind of a cheesy plug for insitro, of course. So insitro, we were founded about 4 years ago. The whole vision being to really merge machine learning with drug discovery, which is kind of a pretty big task. There's a lot of components to drug discovery, and machine learning is a constantly growing and complicated field now. But really, we're just kind of taking the view that at this point, biotech has gotten to the point where we can produce massive amounts of data, really high quality, high content, and high throughput data using next-gen sequencing and single-cell RNA-seq and high content imaging and all these other high throughput biology techniques. But it's getting to the point where, I can't analyze the data. One person can't really just go through it and create really good ideas from that. So really, machine learning is a great way to dig into massive data sets like this and start to kind of unravel some complex biology. And so insitro was founded on that vision, and it's really that convergence of machine learning, computational biology techniques, as well as high throughput biology that allows me to present kind of what I'm going to describe today as our POSH platform. Okay, so I'm going to start with a kind of a very high level view of machine learning. It's, as I mentioned, there's many aspects to it these days, and so there's a new paper on machine learning pretty much every 5 minutes, I feel like, at this point. But at the base, you can consider machine learning to be a way of basically training these algorithms where you have tons of data sets that you can feed into the algorithm to train it. So it starts to learn patterns within the data that you're feeding into it so that it can start to make really powerful predictions at the end. So for a classic example, and machine learning engineers always love using cats and dogs for some reason. I agree with it. They're adorable. So they're feeding in, thousands and thousands of pictures of dogs, thousands and thousands of pictures of cats. Eventually, the model, using a lot of different methods, this one I'm describing here is just a convolutional neural net. It breaks down the image into these components and eventually starts to make predictions. So when you start to feed it images of dogs that it hasn't seen before, it can still pretty confidently tell you, hey, not only is this a dog, it's probably a Samoyed, maybe a Pomeranian. So you can really get complex predictions from these algorithms. And that's just using dogs and cats. Humans are already pretty good at identifying animals for obvious reasons, evolutionarily, you could say. But you can also feed other types of data. So it doesn't have to be imaging data. It could be DNA sequences, it could be audio, it could be natural language processing. There's all kinds of different data formats that you can feed into these algorithms now. Fortunately for this talk, which we will be referring mostly to images for the stuff that I'll be referring to. But one of the tricky parts for all these, that we're really learning from machine learning algorithms is that they are only as good as the data they're trained on. So if you feed, it's the classic garbage in, garbage out. If you're feeding a model a very small amount of data, it can't really predict accurately if you start to ask it a question about something that it hasn't seen before. Additionally, if you start to feed it mislabeled data, so say our cats and dogs, we have a few pictures of dogs in there that are labeled as cats, that's going to start messing with your algorithm as well. So it's really important to produce massive amounts of accurately labeled and high quality data that you can feed into the models. Some of the best models that we've seen, of course, are basically trained on the entire internet of images. And so I think a lot of folks have probably seen demonstrations of that from some of the big tech companies. And this is why a lot of these tech companies are really focused on building up massive databases of just data that they train these models on. So how do we, as a scrappy little biotech, build this massive database of images and just data that we can then feed into these machine learning models? So the approach that we've taken, one of many, is the pooled screening methods. So this is, you can kind of think of this as the converse to arrayed screening, where every condition is in a well. This is still a very good way to do things. We still do tons of arrayed screening, of course. So every, you have a 96 well plate or a 384 well plate, 15, 34, and you have a different drug that goes in every well, or different cell type, or different knockout, whatever your assay is. That's a traditional arrayed screen. In pooled screening, usually you have some kind of barcoding technique that you're applying. You mix it all together, you treat it all as one, and in the end, you usually read out that barcode to identify basically the different conditions within a pool. So this is the way one of the ways we've taken to produce this massively scaled, really high diversity data sets. And one of the nice perks to pooled screening is you also are able to remove a lot of artifacts. So well-based screening, it's great, but you often get some of these evaporation artifacts, slightly different treatment amounts in every well. All these things can produce artifacts, and machine learning models are famously good at using artifacts to cheat. So if, say, you had a little watermark in every image, it is totally just going to look at that watermark and make the prediction based on that rather than based on real biology. Okay, cool. Let me move this over here real quick. All right, so let's get into really kind of the basics. I'm going to start with some, hold on. Sorry, had a little glitch there. All right, we're good. So I'm going to start just with the basics to get everyone at the same point here. So this is basically a quick introduction to CRISPR screening. All right, so I'll start with just CRISPR itself. So in the case of CRISPR, you have your parental cell line. So this is our wild type controls, you could say. All we're doing is we're designing a guide RNA. So this guide RNA is going to basically tell Cas9 where it's going to cut in the genome. So say we're interested in knocking out gene X, we design a guide RNA that's going to target gene X, attach it to Cas9, and then transfect the cells. And basically what you get is Cas9 is going to bind to that guide RNA, find that spot in the genome, and make its cuts. It's going to make an insertion or a deletion, which is generally going to scramble that gene's function in that spot. So when you do that, it's not 100%. You end up with a mix. We're getting better and better at getting pretty high percentages of cutting in these pooled populations. But usually you have some cells that just didn't get cut, and most of the cells did usually get cut at that site. From there, usually you subclone it out. So you take that pool of cells, you seed them into individual wells, grow them back up, and then you end up with, some of those wells were expanded from a knocked out cell, and some of them were expanded from a wild type cell. So you have your knockout and your control. And from there, you can do whatever kind of profiling you want to do. Say you're interested in the transcriptomic effect of knocking out gene X. You, of course, would do maybe singles RNA-seq or bulk RNA-seq. You could do image profiling. Say you want to look at, I don't know, how it affects the migration of macrophage when you knock out the certain gene. You do whatever assay you want at that point. So basically, we know what gene we want to look at. We're going to knock it out and then see how that affects things downstream. So that's kind of your classic. This is a classic CRISPR experiment, and this is done a lot, and it's a super effective way of digging into the kind of specific biologies of one gene. CRISPR screening, it kind of takes it and it reverses it a bit. So say we're more interested, say we have built a really good assay, or we have a disease biomarker, for example, or just any protein of interest, and we want to see what are all the genes that we can affect upstream of that are going to alter that phenotype or that disease. So I have just a little toy example here. I randomly picked a protein, so say cardiac troponin T. So say we're asking the question, what are the genes that are upstream of cardiac troponin T that when we alter them, it leads to increases or decreases of cardiac troponin T in our cellular assay? So it kind of starts with, I like to think, kind of two ingredients. You start with your cells that are expressing Cas9. So usually we just have a kind of a KEGG-promoted Cas9 with a T2A cleaving site, which separates Cas9 from blastocyte and resistance gene. this allows us to put blastocytin into the cell culture to kill off cells that haven't received the Cas9 transgene. So basically, you can be pretty confident at this point that every cell is constitutively expressing Cas9 inside the cell. We then choose the genes that we want to screen on. This could be, a very focused set of, maybe just 100 genes within a pathway, the pathway of interest that we're looking at. This could be druggable genome, which is usually 1,000 to 2,000 genes that we know either have a good small molecule inhibitor or are likely to be able to design one. Or you could go whole genome. If you have the bandwidth and the capacity and the incubator space, you could do all 20,000 genes. Obviously, if you have the choice and you have the room to do a whole genome, that really is exploring the entire array of possible genes that affect this gene. So that's the best way to go. But again, it's huge. That's a huge experiment. So those are the two starting points. Once we have our genes chosen that we want to screen on, we basically design our guide RNA. So those are those little things that are going to tell Cas9 where to cut. So at insitro, we've basically aggregated kind of a combination of a lot of different guide RNA designs. There's a lot of great academic groups that have used machine learning to actually design some of these guide RNAs. So we aggregate a number of these databases of guide RNAs to select the guide RNAs we're going to use. And then we just clone them right into our lentivirus library. So it's just a nice little backbone that we clone the guide RNAs into. We include puromycin so that we can, once again, select off cells that didn't get infected. So we're only looking at cells that have received a guide RNA. So we've packaged this lentivirus library. So each little lentivirus particle in here is going to contain one of these many, many different guide RNAs in our library. We then infect those Cas9 expressing cells with this pool of lentivirus. So you can imagine I kind of color-coded them to represent this. So every cell in this pool now has received a different knockout. Some of them, every single one of these has a different guide RNA in it, and which leads to a different cut. Okay, one important point when you do any kind of CRISPR screening or pooled, genetic knockouts is you do have this fitness effect that occurs pretty much no matter what. CRISPR-I and CRISPR-A have less of it, but you can basically assume that when you're knocking out a gene, of course, that's critical for cell function and cell survival, also mitosis. If you're screening in a cell line that's dividing, you're going to see effects as well. So basically, you're going to start losing all those genes that are critical for cell function. While on the other hand, any genes that are more repressing cell division or cell health, the p53 pathway always kind of comes up. All these other kind of cancer-linked genes, when you knock those out, those cells are going to probably divide a little bit faster and start to take over the culture. So you always get this fitness effect. So it's really important when doing a screen like this that you don't passage these over and over and over again, because eventually this pool will once again become just a pool of p53 mutants or whatever, a mixture of the cancer-linked gene knockouts. So keep that in mind. We also use that fitness effect. One can screen actually on the fitness effect itself. This is pretty common. Or you can use the fitness effect as kind of QC. So you should expect a loss of some of those functional genes. And it's a nice way of knowing actually that your screen is working. Great. Okay. So now let's look back at, remember the original question. We want to find out what are the genes we can knock out upstream that lead to cardiac troponin T upregulation or downregulation. So you got this pool of cells. They've all got their knockout. They're now expressing variable levels of cardiac troponin T based on the knockout they received. And now the traditional way to do a CRISPR screen is to now FACS sort them usually. So you're going to dissociate them. Say you have a reporter for cardiac troponin T, or you can put on an antibody for cardiac troponin T, whatever you need to read out that level. And you do a sort. So you kind of just bin them. These are the ones that are higher than average. These are the ones that are lower than average. You can also do 4 bins, 6 bins, however many. It depends on your cytometer to get more granularity of these binning. But basically, you're separating the high from the low. Once you've done that, remember, this is the pool of cells that has high cardiac troponin T levels. You then extract the genomic DNA and amplify out this guide RNA sequence within this pool. And then you do the same over here and take out the genomic DNA and look for the guides in that pool. And what you really are looking for here is the relative amount of each guide in this pool compared to this. So you can assume, say, there's a guide RNA in this pool that's really common. So a certain knockout that really, they seem to always have high cardiac troponin T levels. When you combine the guide RNAs and start to do some statistics, you can get to the point where you can make the conclusion, hey, knocking out that gene leads to increased cardiac troponin T levels. Conversely, looking at guide RNAs that are in this group, of course, if there's something that's really common on this side, you can assume that maybe knocking out that gene leads to repressed levels of cardiac troponin T. One thing, of course, in this screen I would include as, of course, a positive control is cardiac troponin T, the gene that encodes for the protein itself. So I would throw that in there. You would expect once you make the pool, once you sort it, of course, cells that receive that guide RNA should have very low levels of correct component D. So that would be a nice, just a quick check to make sure your screen worked, for example. Okay, so this is kind of a traditional CRISPR screen. It's super powerful, and you can really uncover entire pathways using this method. And yeah, they're really effective. You can screen on all kinds of things. Really, if you have a protein of interest looking for things that go up or down, this is really a good way to go. You can get really high scale using this. Okay, but we kind of raised the question, are there ways we can do CRISPR screens on more than just maybe protein up, protein down, or RNA up, RNA down, a single thing that you're sorting and binning on? Is there a way that we can make really kind of high throughput, high content data, but still have that diversity and the scale that you can achieve with a CRISPR screen? Additionally, we also kind of wanted to build a platform where we can do more, kind of expand the range of things you can do a CRISPR screen on. So this is, again, protein up, protein down, but there's all kinds of interesting biology, things like cell migration, neuron firing, protein aggregation, protein mislocalization, where you wouldn't really be able to pick them up with a flow screen, but you can pick them up with imaging assays. Because if you could expand this kind of screen into more imaging assays, it really opens the door to more thousands of different kinds of assays that you could potentially do a CRISPR screen on. And so I present what we call the POSH platform. So this POSH stands for Pooled Optical Screening in Human Cells. It doesn't necessarily have to be human cells, but POSH just sounds fancy. It's got a ring to it, so we added the H in there. So it could be whatever species you want to do a screen on. So yeah, I do want to first give a shout out to the Key et al. Paper. That group did a lot of work, developing the rolling circle amplification, which is really critical for this technology, as well as David Feldman, who joined insitro as a co-op for a while to help us build the platform, and the Blaney lab that's done a lot of really important work in this field as well as some of the other. This is a pretty new type of screen that's just evolving, but there's a lot of really great academic labs, kind of that are starting to explore this technology. I want to give a shout out to all the really great academic work that's been conducted so far in this field. So stay tuned. I think keep an eye on the Blaney lab for sure, as well as several other POSH screening groups. Okay, so the way this works, it's a little bit more complicated, I say. So we're starting with that engineered cell pool. So remember, going back, we have that guide RNA library, we have the Cas9 expressing cells, we mix them together, and now we have a pool of cells that have received every knockout, kind of all in this big old well. At this point, you can do really any live imaging assay. So there's a ton that we've now tried, and done a little bit of changing to make them compatible with POSH. And then, so you do your live imaging assay, get whatever data you want for whether it's reporter localization or cells migrating or interacting with each other. We then fix them and permeabilize them, and then we do basically our fixed assay. So we've done kind of a modification of cell paint. We call it morpho paint, which is very cheesy. But it's basically adapted very similar to cell paint, which is staining various organelles, but we made it a little bit more compatible with POSH. As well as, you can throw in antibody staining, mRNA fish, all kinds of, and look at splicing, whatever assay you want on the fixed cells. And then comes the POSH step. So basically think of this first step as the cell culture, running your assays, a little bit of modification to make sure they're compatible, but usually everything we've tried so far has worked. And then the next step is kind of the molecular biology component, which is really reading out the guide RNAs that were in every cell. Okay, so we have this data. This is our phenotype data. This is going to tell us what guide RNAs are in there. So let's zoom in on one of these cells. So this blue guy over here, we're looking at basically the POL2 transcripts. So in that vector that we've infected in every cell, basically there's a POL2 transcript which encodes the BFP of puromycin, and which is encoding the proteins that we need to help our library function. And then there's the barcode, the guide RNA is kind of tagged onto the end. And that's not actually doing anything. We also have a POL3 transcript, a POL3 site, which is actually transcribing the barcode, the guide RNA component. But just the fact that this little barcode is tagged onto the POL2 transcript is what we take advantage of. It's just a handy little convenience that allows us to read it out. Okay, so we have this POL2 transcript that's floating around in these cells, puromycin linked to this barcode, this guide RNA that's in there. What we want to do is sequence out that guide RNA, see what's in there. So our first step is just reverse transcription. RNA is going to degrade very quickly, so you really want to reverse transcribe ASAP before the RNA just goes away. So first reverse transcription. The next step is pretty cool. It's called padlock binding. So we have this oligo that's designed, which wraps around this guide RNA sequence. It has homology basically to the 3' and 5' ends of this variable guide RNA sequence, which it's going to hybridize to. It ends up producing this kind of this not yet closed loop, but it flanks the region. The next step is gap fill, ligation, and RNase treatment. So here's the guide RNA here. Gap fill is just filling in the nucleotides that were missing from when we found the padlock. Ligation is going to close that loop to form a nice tight closed loop, and then RNase to degrade the RNA that's still in the well. Okay, and now here's the kind of the secret sauce is well, it's not secret sauce, it's great work by Key et al. But this is like one of the key parts is the rolling circle amplification. So we have this enzyme that we put on there that's basically going to loop around over and over and over within this circle and produce 1,000 copies of this little sequence right in that location. And so this rolling circle amplification happens. You have a very concentrated location where you have many copies of the guide RNAs. And that's what really allows us to image it at this point. So if you were to have just one copy and put on a fluorescent protein that labels like one base pair there before you've amplified it, it's one protein is very hard to image. 1,000 is a lot easier to image with a standard fluorescent microscope. Okay, so we've got 1,000 copies there. What we do next is in situ sequencing by synthesis. So basically what we're doing is putting on these fluorophore tagged nucleotides. So G, T, A, and C all get a different color. Put it on there. It's going to label that first base pair. Then we wash that off, do a bunch of processing, and put on the next one, read out the next base pair, wash it off, next base pair, wash it off, and cycle through this. And what it ends up looking like are basically Christmas lights over here. So we have the red, purple, cyan, kind of go through all the colors. And what it's doing is it's spelling out the guide RNA that's within this pool, which, yeah, it's a ton of work. All of this is of course, a huge amount of work. But the cool thing is this ends up producing an absolutely massive amount of data. So this is all done and this full plate scale. So what we're end up producing is millions of cells where we've done this, whatever assay it is, it's pretty flexible, really mostly any imaging assay. And then we can also read out the guide RNAs that are affecting the cellular behavior in these pools. Okay, so that's the wet lab part. Now I'm gonna kind of jump a little bit between wet lab and dry lab a little bit because it does require a lot of both to get these screens to really work effectively. So on the dry lab side, and I want to give a huge shout out to my amazing computational colleagues that have led a lot of this effort as well. So at the dry lab, remember, we've produced these data sets. So we have what we call kind of our readout image sets. So this can be, again, I'm going to just call it cell paint, the morpho paint, the fluorescent imaging phenotyping data that we produced. On the other hand, we have all those cycles, those say 13 cycles of sequencing by synthesis, those little dots that are spelling out the guide RNAs. So those are the two data sets. First thing you want to do, of course, is just register them. So we're very careful. Our microscopes are great and very precise, but if you take a plate off of a microscope, put it back on, it's going to be off by a couple microns every time. So one thing you need to do is, of course, just register. We include like a host or DAPI stain at every phase just to make sure, and that we kind of use that as our ground truth to allow us to register. So what we end up producing is basically this translation matrix, just this team matrix which applies to every field that we're including, which allows them to adjust a little bit to overlap in the database. Okay, now that all the images are aligned properly and we know everything's in the right spot, the next thing we do is our barcode sequencing. And the first step here is just identifying the dots themselves. So you can kind of use just like a standard blob detector. There's a lot of algorithms to do that. Other ways you could use, we've tried a mix, is using like a fully convolutional network. So an FCN, it's pretty good for segmentation of particles. But there's, of course, a lot of techniques for identifying dots within an image. This is a classic computational problem. So we've identified the dots. The next thing we want to do is really read out what is the guide RNA. So we go image by image, basically looking at the relative intensities of the fluorescence and spell out our guide RNA within these images. So one of the things we do in the guide RNA design part, which really pays off at this stage, is the Hamming distance optimization. So the Hamming distance is basically, say you have your 13 base pair sequence. If you were to adjust one of those T's to an A, you're basically changing the Hamming distance of that sequence by 1. If you were to change 2 of the nucleotides, say the T to an A and a G to a C, now your Hamming distance is 2 from the original transcript. We set it to be 3. The reason we do that is that if, say, we had one bad cycle and we weren't very confident in the nucleotide that we called in that cycle, we can actually correct it. So it's off by 1, but we still know the closest guide RNA to that one. We can correct it, and we're pretty confident that that is the right guide RNA. If your Hamming distance is shorter, say Hamming distance of 2, and you have that one mismatch, you don't know which guide RNA it's supposed to be. There's usually a couple that readout actually could be linked to, so you just have to throw away the data. If it's a Hamming distance of 1, of course, that means that mismatch is actually going to spell probably a different guide RNA, which would not be ideal. It'll give you kind of mislabeled data. So we want to be pretty careful not to have mislabeled data, which is why we do the Hamming distance of 3. One of the things, setting the Hamming distance is tricky, though, in guide RNA design, because it does start to narrow down your selection of guide RNA. So this is kind of one of the cool examples of, on the computational side, it's really nice to have a high Hamming distance, but it also is going to be a limitation on the biology side. So reaching that balance is a nice example of kind of, the computational and the biology kind of coming together and finding a nice point where we can both function really well with the Hamming distance of 3. Okay, so we've done that. We know the guide RNA sequences. One of the next things we do, of course, is just segmentation. For this, you can use, there's a lot of machine learning algorithms now that are coming out for segmentation that are really good. They tend to be overperforming kind of classical methods these days. So one of the good ways, it's published, feel free to check it out, is the feature pyramid network. But there's a lot of different ways you could do the segmentation, but ML is very good for that. So we have our cells segmented, we have the nuclei segmented. Next step is pretty straightforward. We just assign the guide RNAs to every cell. We then also do, just kind of some standard illumination preprocessing, just getting rid of vignetting or any other illumination artifacts, any optical artifact that might be in the imaging run. Usually this is pretty minimal. All right, at this point, we have all this data, and it kind of feeds together to produce these single cell tiles. So it's just a ton of images of little cells, each of which we know basically the guide RNA that's in each cell. And from here, the analysis becomes kind of an open field, sometimes an overwhelmingly open field. There's all kinds of different analysis methods one can use at this point, and I'll cover a little bit later in the talk. Okay, so a lot of people ask me okay, what does the data actually look like at the end of all of the stuff I just talked about? It looks kind of like this. So we have basically this complete profile for a cell. So here's a mugshot of this cute little cell. We have the plate number, of course, the well number, the location, of course, of the cell inside of the well. We also have, of course, information about the neighboring cells. So unlike most screening methods where you have to dissociate them, here we still have that spatial information, so we know the neighboring cells, we know the features of those neighboring cells. So if you're interested in maybe things, cell-cell interactions and like how is the secretion of some protein affecting neighboring cells, you can actually kind of dig into that. But for this one cell, we have so much information that we've accumulated. So of course we have the organelles, we have the nuclei, the WGA with Floydin, all the mitochondria, all these different stains that we've applied, as well as, so we have that raw data, which is often what we use to feed into our deep learning models. We also have generated featureized data. So this is just quantitative intensities, localizations, textures of all these different channels. You also have the correlations. So say you're getting a lot of overlap of Golgi to mitochondria or something, you can start to see those trends with which we extract as features as well. A lot of this is kind of standard stuff. Folks are used to cell profiler. It's kind of the kind of stuff that can kind of get extracted by pipelines like that. And then of course we have the segmentation. So this is more like morphological or like shape specifically features. So cell size, eccentricity, nuclear size, nuclear location, all that stuff feeds into there. There's also all the live stuff that we've done. In this example, I don't have any live data. We've done a lot of stuff where we've produced like videos. You can actually feed raw data of videos into this matrix as well. And lastly, and perhaps most importantly, we have that guide RNA information. So we have the dots, we have the sequence, we also have the gene knockout itself in this data set. So we've got that. That's just for one cell. But of course, remember, we've done this. This can be scaled very easily. So you can do this for millions of cells, and you end up producing basically a really massive pile of data, which is labeled by the mutation each cell received. So it's a pretty new type of data set. Analysis methods are we're trying our best to figure out the best way to dig into it. I think we've made a lot of cool progress, but this kind of reminds me of the early days of like DropSeq and InDrops and other fluidic single-cell RNA-seq data, where we were just throwing t-SNE, throwing the data into t-SNEs, and it's like, this is looking very promising, but I have no idea how to dig into it. Obviously, that field has matured, and I think the optical field is going to probably follow suit in a similar way. Broadly speaking, the analysis that we've conducted has been, you can kind of bin it into two main types. So one way is more feature-based. So it's talking about all the, basically the cell profiler like features that one could extract. This ends up creating this, 1 million cell by 2,000 feature matrix, which is very similar to what single-cell RNA-seq data looks like. Conversely, one could really take more of that raw data and just feed it into deep learning models. I'm going to talk a little bit less about that. One thing that I have found is that basically the deep learning models start to outperform the feature-based methods if you're starting to get really high coverage of every guide RNA in your pool. So when you start, not surprisingly, as you accumulate massive amounts of data, the deep learning models are able to find patterns better than just feature-based methods. But again, we're talking like 800 cells per guide RNA, roughly, is kind of what it's looking like, but it's too early to really say that. But those are the two methods that I'm going to bin. I'm going to talk mostly about the feature-based stuff for now, just because it's a little bit more established. But there's a lot of ways to dig into this data, for sure. Okay, one of the QC is never the most exciting thing in the world, but it's very important. So, I want to bring it back to, insitro's goal is to find good targets. We want to make drugs for patients. And so we really want to make sure we're not messing anything up along the way. It's got to be high quality data all the way through. So we're not introducing artifacts. We're not losing stuff that could really help us along the way. So there's all kinds of QC that we've implemented along the way, and anyone that's doing these kind of screens, I would highly recommend trying to implement a few of these themselves, because it really helps just to keep an eye on it and make sure everything's working well. So the first one over here, we just call it sequencing signal to total ratio. It's really straightforward. We're just taking those little dots that we're imaging. You say we called it as a T, so the intensity of the T fluorophore is the highest. It's basically I sub T divided by I sub A plus I sub T plus I sub G plus I sub C. You're basically taking that intensity of the highest one and making sure it's way higher than the sum of all the other ones, including the T itself. So it should always be, of course, less than 1, but you do expect it to be, somewhere in the 0.8 range is usually what we shoot for. We can actually still make the call pretty confidently, even if it starts to drop. But again, then you're starting to depend a little bit more on that Hamming distance correction. You're going to start to lose guide RNAs, and you might even potentially start miscalling guides. So you want to keep that nice and high throughout a screen. The other thing I like to do is, it's really just a nice way to visualize it, if anything, is to take all those dots. You basically have 4 values, the G, T, C, and A. Just do a UMAP of that, and you can start to see if any of the different fluorophores are starting to merge. So you get really good separation. It looks like worms for some weird reason. So obviously it's really good at just the G is out there, the A is out there. The 2 nucleotides that do seem to get mixed up is C and T. So you notice there's a few that get called as a C, right? If you see it right there, it's a tiny bit of dots, which are actually T's. And then if you look at this plot, you can start to see if this is starting to happen. So you should raise some flags. But fortunately, if you look at the Hamming distance correction equivalent of this plot, so take these same dots, but look at them after the correction, those actually get turned back to T. So we are able to account for any of those fortunately. But again, you can only handle so much error correction with the Hamming distance, so you don't want to rely on that too much. All right, one last thing that we run is basically looking at the fitness effects. So I mentioned you're going to have some guide RNAs are going to die off, some are going to take over. With any CRISPR screen, it's usually a good idea to extract genomic DNA along the way, sequence it out to see that distribution of guides. Usually, you will see, this is typical and expected. You'll see the loss of certain guide RNAs. So these are our fitness ones. What we do with POSH is we take the very final time point. So basically right before you fix the cells, we extract genomic DNA. We look at the distribution of the guide RNAs in that data. And then after we've done the POSH processing, we then look at the distribution of guide RNAs in that data. And you would expect they should be pretty much equivalent. If there's something wrong with your processing, you're selecting for certain guide RNAs, or there's you lose anything that's GC rich, and specifically in your POSH sequencing or something weird like that, you would expect that to be really variable. But fortunately, in this case, or so far, we've seen usually pretty good agreement of our genomic DNA to our POSH readouts. Okay, so that's kind of walking through a little bit of the platform and all the technical aspects. I do want to pause here to see if anyone has any questions before I get into some of the data analysis. Let me make sure I can see if anyone has any. Otherwise, I'll happy to move along. Nope. Okay. Cool.
Nope. Yeah, I don't see any in the chat. I think you're good. This is
right.
Very thorough. I really appreciate you characterizing the different QC elements as well. I have high confidence in the data that we're about to look at next.
I know it's important. QC is not very exciting, but it's important.
It's exciting for people who are good data stewards, though, right? Like if you're working with data and the QC's off, you can't trust anything, and it's hugely important. Yeah,
exactly.
Yeah.
Great. All right. Cool. So I think everyone's probably excited to see what this data actually looks like after all this description of the platform. Okay. So I'm just going to go over just a couple really case studies of some screens that we've done. So there are more pilots just to demonstrate the technology, and then some of them are digging into some pretty exciting biology. Okay, so this is just one example. This is our A549. This is a cancer line, very well-behaved cancer line. It's really nice to work with them. They don't differentiate like stem cells, so it's kind of nice. And so the assay we did here was MorphoPaint, which is very similar to cell paint. Again, just slightly modified so that we can it's compatible with Posh. Okay, so the guide RNAs that we applied here, we basically selected a number of genes that we know are going to be probably show very similar morphological phenotypes. So we took a subset of like 20 to 30 mitochondrial translation genes, a subset of 20 to 30 proteasome-linked genes, a subset of 20 to 30 actin-kinesin genes, and designed a bunch of guide RNAs for those genes and put them all in this pool, with just to test whether those genes do end up getting predicted to be similar in their behavior. So we started with, we ran the screen all the way through. You end up with basically a stack. You remember those little profiles of individual cells. And we end up with a stack of 286,000 cell profiles like this. Next step, very straightforward. We just do, again, this is pooled screening, which allows you to do some pretty nice normalization of the data. So one thing you can just do is well-wise normalization. You have every condition actually within every well. So this normalization will get rid of any artifacts that might still be in there. So this is a nice starting point. The data ends up being very clean with this normalization. Then we do pretty classic just principal component analysis. We usually just bring it down to like 30 principal components, seems to do the trick, and cover the vast majority of the variability in the data. All right, so the next thing we do, so we've got our principal components. Now we usually aggregate the cells by the guide RNA. So we have, say 1,000 cells that received every guide RNA. You're going to aggregate that data, kind of end up producing kind of almost a signature of every guide RNA in your data. And we start to calculate the distances between those guide RNAs from each other. Now I can break those distances into kind of two things. And this first one is surprisingly useful. And I kind of call it the intergene guide RNA, which is basically given certain genes, so say some mitochondrial translation gene. So we have 7 guide RNAs that are all targeting that gene. We look at the distances between those guide RNAs that should have the same effect on that gene, or in the cell. So if it's a strong phenotype and the guide RNAs are all working, you would expect it to have a pretty distinct phenotype, and the distances between those guide RNAs should be pretty small compared to the general data, the latent space of all the data. So if they're all agreeing, if they're all kind of reach a consensus, if you will, you can be pretty confident that that's a real phenotype from the gene that you're knocking out. If you have, say, of those 7, maybe 6 of them are all agreeing, and then one of them is just hanging out in intergenic control or non-targeting control space or some other guide RNA, then those 6 are probably right, and that one guide RNA that is disagreeing with the rest of them it indicates that guide RNA is probably not targeting the right spot, or it's just not cutting. Lastly, sometimes in a lot of cases, you just won't see a strong phenotype. So it's kind of a spread. None of the guide RNAs are agreeing on a phenotype. They're maybe all kind of similar to non-target or intergenic controls. In that case, you can probably assume that knocking out the gene isn't really having much of an effect in this assay, in this cell culture. Or maybe you have just too low of a coverage. You need to increase your statistics to start to see a phenotype. Okay, so that's just making sure, that's kind of identifying which guide RNAs or which genes are leading to phenotypes. Of course, the other thing that we do, and this is what really generates the interesting, maps of biology that you can produce with these screens, is looking at the intra-gene guide RNA. So we're looking at guide RNAs that are targeting different genes and starting to see what are the distances there. And that's where you can start to build these maps of guilt by association, is kind of how I like to refer to this analysis, where you're finding whole groups that all seem to have similar behaviors and start to make conclusions from that. Okay, so this is kind of what the data looks like. It gets a little, similarity matrices can always get a little bit out of hand if I start plotting it, especially at the guide RNA level. So on the right here, we have basically the similarity matrix of the genes, so aggregated to the gene level, and then the distances between those genes. And then on the left here, I have at the guide RNA level, this is just a spring forest diagram network plot. And with these plots, you can kind of show guide RNAs or larger numbers of nodes more effectively. So basically on the right, you can see we did hierarchical clustering of basically these genes, looking at what are similar to each other. And just at a glance, you can see all the proteasome. And this is sorry for the tiny font. So this is all proteasome. We have microtubules, dyneins, mitochondrial translation there's a couple main clusters, as well as one very core cluster here. Actin kinesin forms a group down here. So we're definitely getting clustering of these genes by the global morphological effect. One thing I wanted to note, remember, I haven't really put in necessarily an assay that's going to test us for actin/kinesin or Golgi ER or proteasome necessarily. We're just throwing on cell paint and kind of letting the genes do their thing and seeing if any of these many morphological assays we put on there are showing phenotypes. So you don't necessarily have to go in with a hypothesis for this kind of screen, where this is very exploratory the way this screen was run. Okay, so that's what it looks like in the similarity matrix. If you look at more of the network plot, you can see, we have, and I definitely could not label every individual gene in here, but basically there's a giant mitochondrial translation cluster. The controls kind of form this spread group here, and then the proteasomes regulating guide RNAs are right there, Golgi IR grouping up right there. But one of the really neat things is some of the guide RNAs were really out doing their own thing. So you see RAC1, this is the 877 guide RNAs all targeting RAC1. They're all doing something very different from the rest of the guide RNAs. So that's kind of fun. TPT1, similarly, it's doing its own thing. Actin B, I'll get into that in just a second, was really interesting, kind of doing its own thing as well. So we're able to, basically start to map out a lot of these, these guide RNA, these gene effects, without really having to feed it any initial hypotheses, any priors. It was just kind of accumulating them based on their similarity using this more high content data that we're feeding to the system. There's one thing to say, yeah, they're clustering. It looks good, but of course I wanted to quantify this. So what I did was use StringDB, which is a really great resource for folks that are familiar with it. Basically, it's an algorithm that was developed that kind of accumulates, whatever 100 years of literature, and it's just a giant literature scrubber that has really made these really impressive, gene-gene interaction networks. Basically, a big map of genes and proteins that are basically linked to each other or related to each other. So what I did was take our list of genes and I applied that to StringDB, which made a very similar looking network, and just basically filtered on really strong edges between genes in StringDB and looked at what is our score compared to those. And there's a very clear correlation where if StringDB is calling a really good edge between any two nodes, this one morphological screen is able to also call very, very high similarities between those genes as well. So it's a nice quantitative check to make sure our model is really predicting connections as well as we hope. Okay, so one of the really neat things about this screen, I didn't even think about it when we were setting it up, but it's really nice compared to our classic CRISPR screens. So remember, in a traditional CRISPR screen, usually you're binning the cells in some way, and then you're dissociating or breaking apart the protein, proteinase K, and then you're just extracting the genomic DNA. And then you're looking for the relative amount of guide RNA in each group, and then you have your hits, but then you have to do follow-up experiments to really validate your hits. In the case of POSH, I would still recommend always validate your results. But the nice thing about these experiments is we still have the data. We have the imaging data. We can just basically control F for the knockouts that we find, any of the hits that look promising, and just look up the images of the cells. It's of course a little bit more code than control F, but basically find the knockouts within the data, just to validate and make sure what you're seeing is real. So I'm going to go through just a few examples here. So that mitochondrial translational core. So this is there's many genes that fell out. I just kind of picked one at random. So mitochondrial ribosomal protein L57. So this clusters tightly within the rest of those mitochondrial translational genes. basically the first thing I did was kind of differential expression analysis, but not on transcripts, which is what it's traditionally used on. We used it on these morphological features, and the features that stood out are, not surprising, mitochondrial features. So this is generally just paranuclear cytoplasmic mitochondria intensity, and other aspects are all kind of groups up. You can see and kind of are way different from the intergenic controls here. You can also take, one thing we do is just take the individual, choose a few of these features, and just look at how the distribution, you can make histograms of all these quantitative things, because it's a ton of data, and look at how that compares to intergenic. And sure enough, there's a big drop in the mitochondrial levels. This is specifically looking at the cytoplasmic variants of mitochondria, of mitoprobe. And then, of course, the best part is you can actually just look at the images. So here's, what are kind of our morpho paint. Again, you can just think of it as kind of like cell paint. And then specifically looking at the mitochondria, you can see this cell has much lower mitochondria. One cool thing, it is a pooled screen, so all the cells right next to it have totally normal mitochondria. So it gives you a lot of confidence this is really a real phenotype you're seeing there. So that's nice. One of the really fun stories was, beta-actin. So ACTB, you notice it kind of stood out a little bit. I did the same differential expression analysis and noticed phalloidin was a lot higher in this one. My first instinct was that I just messed up a sign, that I was subtracting instead of adding, or had a minus in there that shouldn't have been in there. Because you're knocking out actin, you should see less phalloidin, presumably. But sure enough, it was higher. Doing the follow-up, looking at the histograms, looking at the images, you have cells that clearly have a lot more actin compared to some of the surrounding cells. The green is phalloidin in this case. Sorry, not to mix it up with the mitochondria over there. And doing some reading, it turns out cells often have or I guess not that surprisingly, cells are really good at compensating their levels of actin. So if you knock out beta-actin in a cell, it can overcompensate many times with gamma-actin expression. So this is pretty common, and there's a paper that's published on this, and I think we accidentally validated those results, and it looks pretty, or by happenstance, validated those results. So it was really neat to dig into some of that biology there. One of the other things one can do, of course, with the follow-up is basically take the transform of that matrix. Rather than looking at the differential expression of features in a single gene, you can take a feature that you're interested in and look at how the different knockouts are affecting that single feature. So I took as an example just nucleus size. Some of the top hits looking at things that increased nucleus size were, not surprisingly, kinesins. So kinesin family member 23. So this is it's a molecular mode. It's part of the contractile ring that forms during mitosis. So cell divides, bipolar spindles pulling your chromosomes apart, and then the contractile ring forms at the cell membrane and closes it and kind of seals it off to make two cells. Without kinesin family 23, that contractile ring is disrupted. So the cell still the nucleus can split, but the cell does not. So you end up with a lot of multinucleated cells. And sure enough, pulling up the image, you can see all these most of these kinesin family 23s have 2, 3, 5 nuclei sometimes in each cell. Kinesin family 11 also was a top hit. This molecular motor was involved with is bipolar spindle assembly, so it is no longer able to pull apart the nucleus very effectively, which often leads to aneuploidy. You still get multinucleation sometimes, but often you just get really enlarged, kind of dysmorphic nuclei, and that's what we were seeing in our images as well. On the opposite side, TALEN1, it's not linked to really nuclei function. It's linked to focal adhesions. So these cells that have this knockout, they're just not sticking down and spreading out because they don't have TALEN1, and so they're kind of lifting up a little bit. The nucleus is getting small and balling up, so that's what gives us that nucleus size feature. So it's interesting to look at the images and do some follow up to see what's really the biology behind some of these hits. Okay. So hopefully the video's working. All right, so I mentioned this is a ton of work. You get a lot of data from one screen, but we really want to scale it up, get to ideally, whole genomes where the story starts to get really, really exciting. So we've implemented automation. This is just one of our little robots we're really working on really at insitro, just scaling up a lot of our assays, not just the Pouch platform, but really across the board. So we've got an awesome automation team that's putting all this stuff together. So not only do we want to increase the scale, but automation also helps to reduce the variability between runs. So we've already can address a lot of the batch artifacts because it's a pooled screen, but still you want it to be really consistent. Machine learning models are going to be able to, cheat with batch effects. And so as we build up these massive databases of images to train our machine learning models, we really want to make sure it's pretty consistent data every time. So automation really helps us with that. Also, eventually we hope to just let the machine do all the work so we can sit on the beach That's might be a little bit more aspirational, but someday that would be delightful. Okay, you can see it's doing a lot of plate switching. The robot is very busy. Cool. All right. So with automation, we can scale this up. So I'm going to talk about kind of our second case study, which is basically taking it to the druggable genome scale. So this screen is very similar. A549's cancer line. Again, we use this morpho paint, but we also added in phospho-S6 antibody. So S6 phosphorylation is a really strong indicator of mTOR activity. If mTOR is high, S6 will get phosphorylated typically. And it's there's really good antibodies for it. So you can just kind of use that as a measurement of mTOR activity. So we have we include the druggable genome library. So this is 1,722 genes. We added in some more core mTOR pathway genes. We just kind of wanted to give an example of a pathway that we wanted to kind of do a little bit more of a deep dive into, just to make sure everything's working. So we went with the mTOR pathway is near and dear to my heart, so we went with that one, as well as a number of course, non-targeting intergenic guide RNAs just as our controls. Okay, for this screen, and this screen really ended up being, I think, just one. I can't remember if it was like one or somewhere between 1 or 2 plates, not a ton of plates, but what we ended up producing was 1.7 million of these cell profiles within this experiment. So very similar process, normalized based on the well. We ran principal component analysis. There's a few other, of course, little technical things we do in here, but basically this is the mainstream of the data. Overall, this looks a lot like the previous one I showed. The one thing I want to highlight is that we generally aggregated to the guide RNA and to the gene level a little bit earlier, because even the beefiest of EC2 instances starts to struggle when you're dealing with a 1.7 million by 2000 something matrix. So the aggregation is a little bit different for when data sets reach this scale, but so far we can handle it. Yeah, so the first thing we did to check was look at that. We had that S6, and this is a little bit more like a traditional CRISPR screen, just looking at S6 phosphorylation and looking at how the knockouts affected that. One of the main differences here is that since we did POSH for this, we still have single cell readouts. So rather than just a bin of this, positive versus negative, we do still have those histograms of phospho-S6 levels. So we have single cell level data, which does give you a lot more statistical confidence in your hits, which is really nice. So not surprisingly, RPS6KB1, this is the kinase that sits right upstream of S6, which phosphorylates it. That was our top hit. So that's really nice to see, of course. TSC1 and TSC2, these are the genes that cause tuberous sclerosis complex. So loss of function of TSC1 and TSC2. Basically, it takes the foot off the brakes on the mTOR pathway, and it leads to really hyperactive mTOR function, which thus leads to really high S6 phosphorylation. So those are all of our top hits. This is kind of the who's who of the mTOR pathway that came out in this hit. So a nice validation that it works, and that single cell level resolution gives us really, really nice statistical confidence in some of these hits. All right, but I also wanted to ask, what happens when we apply that hypothesis-free, throw all the data at the algorithm and see how it starts to map these things out. Again, this is just like 1 plate to 2 plates of data. It's still a ton of work because all the work that has to go into the molecular biology and the sequencing part. But you can think of scaling this even further in a pretty straightforward way. So this is our 1,700 gene data set, very similar to that map I showed before. I'm going to take a moment to go through it because it's a lot, and you guys, hopefully folks can see on their screens. Basically every node here, again, we're taking the guide RNAs, aggregating to the gene level, and then looking at similarities between the genes. Every node here is a gene, which has each of which has multiple guide RNAs. One thing I did, so you remember that guide RNA score where if all the guide RNAs are agreeing on a phenotype, I kind of give them a high guide RNA score. I represent that here in this plot with a large dot. So if you see a gene with a large dot, you can be pretty confident that's a real phenotype that's kind of standing out from the rest. And so it gives you more confidence that it's probably a real hit. If the dot is a little smaller, it usually means we're less confident in it. It doesn't mean it's not real. It just probably means that we could probably boost our numbers. If we run the screen a little bit more, maybe throw more guide RNAs, we can start to see if it's real or not. So it gives a relative confidence value in the map that we're forming. And then, of course, the edges. You see, we formed edges if there's a pretty high similarity between any 2 genes. If it's a really confident, like these are identical almost, we put a very kind of a thicker line. You can see this case is, I think, a perfect example is TGF-beta receptor 1, 2, 3, and SMAD3. These are the kind of the core genes in the TGF-beta pathway. Those all have very very high similarity and very thick lines connecting all those nodes. So that's a nice example of really high confidence. If it is like a thinner line, that still means they're similar, but just again, relative amounts of confidence between those. Okay, so this is a huge map. Obviously, I don't have time to go through all of it. I just took a few examples. Again, mTOR is kind of near and dear to my heart, so I'll just zoom in on that one. One of the interesting things is that we included those mitochondrial translation genes in the screen as well, and those tend to like group up with the mTOR quite closely. I guess this isn't too surprising because of crosstalk between mitochondria and mTOR. It's very, very tight. If mTOR is high, mitochondria is high. If mTOR is low, mitochondria tends to also go low. Basically, the cells in active form a lot of protein, but also make a lot of energy mode versus the converse. So those tend to move around a lot together. But basically, we end up with this really nice, this is basically an mTOR low grouping that we get here. You'll notice it's right next to the EGFR, which I thought was very fascinating because EGFR is, while it's considered kind of a separate pathway, it feeds directly into the mTOR pathway through RAS and also as PI3K. So they're very similar, but there's some slight separation there. You also see up here, this other group out here is actually the mTOR high group. So you can kind of, if you plot this over kind of an established mTOR pathway map, we are starting to pick up a lot of the various genes that are within the mTOR pathway, as well as a lot of potential new ones that need a ton of validation to, for us to really confidently say they're new mTOR regulators. But, you can start to really uncover pretty core aspects of biology with that kind of analysis. All right, one other example I'll just jump into is, so I mentioned that TGF-beta, but next to that grouping, there's kind of this interesting integrin group, as well as this cluster here. And looking more closely at this, you can see there's kind of this core of this kind of constellation of genes in here. So I saw NPC1. This one kind of stands out as clearly, lysosomal function, trans-Golgi network. But right next to that one, we found TMEM199, COPE, TMED2, ATP6, AP2. So these are all related generally to basically Golgi ER trafficking, the COP1 and COP2 complex, which is basically the anterograde and retrograde process shuttling of cargo between Golgi and ER, as well as things like cholesterol, LDL receptor recycling and things like that. So these are all known to have pretty similar function, and there's a whole bunch of other genes that we're finding in there that seem to be related to that. So so I could go on forever, digging into individual clusters, but obviously I won't. But it's really exciting to start to see really key aspects of biology. Again, we didn't tell it to look at LDL processing. We were just throwing basically every assay or every stain we could in this big pooled assay and kind of let the data speak for itself in this way and kind of embedding it in this more, this higher representation of the data. So that's kind of what that data looks like. I'm looking at the time. I'm already pretty much at time. So I'm not going to dig into this one, unfortunately, but I'll just, you could apply it to more than just cancer lines. So one thing we've also done is, taking iPSC-derived neurons and looking at, we have a nice disease model of tuberous sclerosis that we've developed in these iPSC-derived neurons. And really, we basically train machine learning models to identify what the disease looks like. And then when you run this kind of morphology screen on those disease cells, you can start to identify what are the genes that are reverting them back to a healthy state. This, of course, is a very clear application for target discovery. And excitedly, we're finding a lot of interesting and promising targets using these kind of screens that we're moving forward into our massive drug discovery pipeline that we're building. So it's super exciting. It can apply to really anything. iPSC-derived cells, of course, lets you target specific tissues that you're really interested in. If there's a disease that affects a certain cell type, I would definitely recommend doing the screen in the cell type if you can make the differentiation, if that's available. So I'll have to skip through that again. Sorry for the time. But yeah, just to summarize what I just talked about. So, machine learning is great. It's a buzzword, but it is a really good buzzword. It works really well when it's applied appropriately, but it does require really high quality, diverse, and ideally labeled data. It doesn't necessarily need labels, but labels are good. So to do this, we built this, what we call the POSH platform. So this is a pooled optical screening in human cells. It lets you do a CRISPR screen and then read out the guide RNAs basically in an imaging assay. We built a bunch of robots that are helping us to run all this and get it to the scale that we need to really get it to the, kind of high level and massive amounts of data to really build these models. And our initial work, mapping these data sets, we're finding that we can start to basically produce clusters of genes that have similar pathways. And you can also start to do, if you combine it with some iPSC-derived disease models, you can start to find new targets for some of these diseases. All right. So most importantly, I want to thank all the incredible people that have done the work. So this is we just had our 4-year anniversary. There's a cake there. First, I want to give a huge shout out to Bobby, who's done a ton of the work, Srinivasan also, as well as Eric Lubeck, who did a lot of the foundational work, and David Fellman, I mentioned already, who did a lot of work getting things established, as well as really the whole team, John B. I shouldn't start listing names because it's going to take forever, but it's really been a huge team effort. I also want to do a shameless plug for insitro. So we're hiring anyone that's out there that watches this. Please use that QR code to check out our website and see what openings we have. Maybe we have some finishing grad students or postdocs in the audience. So yeah, definitely take a look. So thanks.
All right. Excellent. Thank you so much. That was a phenomenal talk and just a tremendously compelling technology too. I'll wait to see if there's any questions in the Q&A, but I'll kick off a couple. Tying into your hiring plug. So you have presented a very interdisciplinary project. There's cell bio, molecular bio, comp bio, plus all of the imaging analysis, of course, deep learning, and robotics. So for the trainees in the audience, what are some of the skill sets that you see as the highest priority for people who are still, in a space where they can really learn easily in universities and whatnot, and they want to build out their skill set?
Yeah, that's a great question. All of the things you listed are great. We will hire any good molecular biologist, computational biologist, automation engineer. They're all very high demand, especially right now. One thing that I found, so as you mentioned, this is a really multidisciplinary project. So our team, and again, I can't give enough accolades for our amazing team. A lot of them are actually hybrid. So we call it wet lab and dry lab, but the term we often use is damp lab. So people that can actually do both. So if you are, it's kind of, I don't know about the term damp lab, but maybe I'll stick with hybrid. So if you're a really good cell biologist, but you're making data, I would recommend, and maybe you're collaborating with a computational biologist, definitely shadow them and see if you can learn some of the analysis. Definitely still work with, if you have some really good computational colleagues, lean on them and use them for advice. But see if you can kind of cross that a little bit. You don't have to be an expert at everything, but just being able to speak across, the disciplines is really, really helpful. We find, especially in companies like insitro that are really kind of an integration of multiple technologies or multiple, disciplines. So I'd say, yeah, branch out a little bit. Obviously being able to code is always very helpful for, I think, anyone. So learn Python. Python is definitely the most popular. But if R is also very good, or whatever you decide to use. But yeah, I think being cross-disciplinary is it's fun. It kind of, there's so much more to learn when you start branching into a new field. So it gets kind of exciting in that regard. So that would be my advice there.
Okay. Yeah, that's a really good point. And I think actually it ties back into the QC too, because when you've sat on both sides of, the lab, so to speak, you ask questions with skepticism in different ways, and you can pick up on things that if you don't have the familiarity in either side, you may not recognize. So I think that's a really good point as well too.
Yeah, absolutely. Yeah.
Okay. One other question I had, and this is more on the technical side now. Are there any biases, and actually it ties in a bit to the QC side, but are there any biases that need to be considered from a data analysis perspective as a result of the rolling circle amplification?
That's a good question. Okay, so maybe if it's more efficient, if different guides, they're going to have all these different sequences. So I'm guessing if maybe one guide RNA has a lot of A's or something, that might affect the rolling circle amplification. That's a good question. We haven't seen it. I think I mentioned that genomic DNA at the very end state versus our POSH representation. So that's a nice way of comparing and making sure we're not seeing too much, and we don't see a ton. One concern I guess I would see is if you do have a repeat of the same one over and over and over again. I'm trying to think if that might affect any of our registration. Registration will be fine. There are maybe some aspects of lead versus lag. So if you sequence, an A then TG, whenever you're doing sequencing by synthesis like this, you have sometimes you can skip one. It's like a 0.5% chance that you're going to skip a base pair, or you might not move on to the next. So there might be a very slight bias based on repetitive aspects of your sequence. But so far, we're seeing pretty comparable representation between the next-gen sequencing method and the POSH sequencing. So we haven't seen that. I'm just trying to brainstorm what are some other ways that could create artifacts there. One of the nice things is that we do the phenotyping generally all before that processing. So it won't. So if anything, it would disrupt maybe the number of guide RNAs or cells with a certain guide RNA we're getting, but it's not going to change the data. Or like the analysis itself. It'll just change the representation, which is you can be more flexible with representation by downsampling or other means.
Right. Okay. Thank you. Awesome. Relating to the technology and the scaling of this, because it seems like there's a tremendous amount of usages. I think, there's of course the applied setting or the applied considerations from a translational perspective in biopharma and biotech, but also it seems like potential to just truly expand our understanding of gene networks. And, so often these things can be very complex and hard to drill in from a reductionist perspective. And I think there's, opportunity from a fundamental molecular biology understanding perspective too. Relating to the technology itself, is this something that in your guys' use cases, you often see like clients or partners come and want to work with you guys and you carry out the experiments and partner with them based around their research questions? Or is this something that you guys envision as like a core capability that you want to expand into a lot of different labs, maybe licensing the technology or what have you?
Yeah, so this is so what we're building right now is mostly there's plenty of demand just within insitro to run so many screens as we're building the platform. So right now I'm just trying to catch up with all the stuff we want to do internally. That said, once we really reach full steam and have this automated more fully, I would love to and we have we have a whole team, and we're very collaborative. We've worked with academic groups in the past, and of course we have big collaborations with pharma. So we're definitely always keeping an eye out for really cool collaborations. You can think of anyone that's developed a really cool imaging-based assay. It's very likely that it could be adapted to pooled optical screening. So I'm assuming a lot of the other groups, I know other groups that are kind of building pooled optical screening platforms are often collaborating with more of a wet lab that's developed an assay to kind of merge them together and see if we can do a CRISPR screen there. So I hope that we can be one of those labs as well in the very near future, I hope. So anyone that's looking that has a really cool assay they want to try, give me a call. We're probably not ready yet, but I think we'll get there at some point. So yeah, we're very collaborative, I think, and we really like working with the greater scientific community on some of these assays.
Excellent. Okay. One of the questions coming in from the Q&A, are there any systems where this works better or worse than others? For example, is Arabidopsis harder? For example, more steps required than, say, E. coli?
So everything we've done that I've described is actually in mammalian cells. So we're not screening in any bacteria. There are definitely cases where it works better than others. I think one of the tricky things is gene silencing is one of the things. And sometimes those dots, it gets harder. For example, I mentioned the neuron screen. So neurons, as they differentiate, or any iPSC model, often as they differentiate, they start silencing different promoters. So making sure that's expressed and continues to be expressed, so you can still read out the guide RNA. So those are some of the tricky things that we found where you got to be careful and do some testing to make sure in your different model it works. But yeah, this is all mammalian. I can't really speak to doing screens, at more in the bacteria level. I guess we do use bacteria, of course, for assembling our lentivirus library and all the cloning that happens upstream. And there, I think I forget which one we use. It's kind of one of the classic, competent E. coli things. One of the classic ones. So haven't played around with that, unfortunately. But yeah, definitely keep an eye on the different models. You got to do tinkering every time just to make sure it works. Yeah.
Right. I'd imagine from like an agricultural perspective, depending on genome size, you could have some differences too, right? As you go into plant genomes, which can be much larger. Yeah.
Absolutely.
Awesome. And I suppose I'll ask one last question, and then we can go ahead and close our session. So in addition to the case studies that you've provided, and I know the last one you didn't have time to go into full detail, but are there any specific areas that you're really excited to apply this technology to? And feel free to expand if it is more that last use case. But just, where do you see this having the biggest impact from a human health perspective?
Yeah, so there's a lot of parts of it that I'm really excited about. One of it is just bringing kind of that unbiased, kind of hypothesis-free kind of mapping that I was showing briefly. That's still only druggable genome. That's only really 8% of the genome. Expanding that to the whole genome, really providing a map of all these genes, more globally and at scale would be very cool. I think that would be really nice. Obviously, there's so much nuance in biology, and it's just so complicated to naively say we're going to make a map of biology. I realize that is optimistic, but producing that kind of map I think you would find a lot of interesting things about various genes if you do that kind of approach. So that I'm really excited about. All these iPSC models, we have a ton of really good iPSC models, so it's really going to be fun, for most of the demonstration, I talked about cancer lines, but every different cell type is probably going to have almost a different map, if you will. So they're all going to behave differently. So I think moving it into different cell types is really going to be fun as well. The neurons, of course, neuron images. I love just taking pictures of neurons always because they're very cool. They're beautiful always. And so, but thinking of other cell types, cardiomyocytes, I don't know, macrophages, microglia. The list goes on. There's so many things where you could really build some of these maps, and I think it'd be really, really exciting. Yep.
Yeah, excellent. Awesome. Well, any last comments from you, Max?
No, I want to thank you again for inviting us. I want to, also doing the industry seminar. It's often, industry is often, you don't hear as much about industry in the public space as much. So having a nice venue for people to see and people that are interested in maybe, making the jump over to industry. Hopefully this is a nice example of, we get to do very cool science in industry, just like in academia. So anyone wants to reach out, again, we're hiring, but just if they have just general questions, I'm always happy to, be in touch. So people feel free to find me on LinkedIn if you have any questions or want to chat. So yeah, thanks again.
Okay. All right, excellent. Well, thank you so much for taking the time to present with us. Fabulous talk. Really enjoyed it. And for those in the audience, the next ISCBacademy Industry Seminar Series will be September 22nd. AstraZeneca and Itai Jacob will be presenting, and registration should be available on ISCB either right now or in the next coming minutes. And if you're catching the recording, it should already be up. So thank you again so much. And with that, we can close our session.